group. He's an associate member, Broad Institute of MIT and Harvard, and he's a principal investigator of computer science and artificial intelligence lab at MIT. Well, he has a list of awards, prizes, numerous. For example, the US Presidential Area Career Award, the Alfred Sloan Foundation Award, NSF Career Award, the AIT Nikkei Award for Science and Engineering, a long list of exceptional research papers, and having a lot of contributions and impact in the area of systems computational biology. He has been doing recently pioneering work in digital cognitive cartography, and he's going to describe about Mantis today in his talk. His group at MIT aims to further our understanding of the human genome by computational integration of large-scale functional and comparative genomics datasets. So today we are going to listen to some of his contributions in this area. Thank you very much for accepting our invitation.
Thank you so much for having me. So again, it's a real pleasure to be here. I've had such an awesome time meeting so many of you, and I'm really very excited about this community. I think that the community of Greek scientists is extraordinary, and I think this space of AI is one where I think we can really flourish, and the per capita impact of Greek scientists in the space of AI is just off scale, extraordinary. So what I'm going to talk to you about today is the concept of cognitive cartography. The idea that we can now start not only talking to the AI, but looking inside its brain. The key idea is that if AI only gives you one answer at a time, if it only gives you one piece of information at a time, if you wish, using a GPS analogy, if it only tells you turn left, turn right, turn left, go straight, turn right, then you can't really inspect it. You can't really choose to take a shortcut. And that's where we are with AI today. We've made extraordinary advances, but we don't have the ability to understand how the AI is thinking. So that's what I mean when I talk about visual AI. What I mean is being able to actually peek inside the brain of the AI, look at a cartography of ideas, look at how concepts are organized, how documents are organized, how collaborators are organized. For example, we have a little WhatsApp group that includes many folks in this group. I'd love to take the descriptions of everyone's work in LinkedIn and then create clusters of people that I should be connecting with each other. Take the individual projects and create another floor of projects and people and how they're connected to each other. Who's following me here? Yes? Awesome. So that's what I'm going to show you today, not just tell you about, but show you. So we basically built this system that we call Mantis, and over here I'm showing you, for example, 20,000 proteins from the human genome organized according to their function and their structure. So now we're able to bring up all kinds of tools. For example, I can bring up the tree, and then this is the ontology of how these proteins relate to each other. You can see here the mini map, which basically allows you to know where you are in the navigation and go through thousands of these proteins according to how they are related to each other. So that's what we want to do with almost anything. In this particular case, these are thousands of patients from the multiple inheritance of man, the mimic data set. And within this, we have an AI system, we have agents that we're able to interact with, we have these tools like cartography that I mentioned, and we're applying this to understand the heterogeneity of patients, to understand the heterogeneity of chemistry and drugs and genes, to understand how to infer the circuitry and the connectivity between all of these different components of the cells. And we're using this for drug discovery. We're using this to understand not just the sequence to structure problem that Demis Hassabis has had an extraordinary impact in and that we're going to hear about in September, but to understand the structure to function problem. In other words, once you have the structure, what good is it if you don't understand how it functions? So that's what we're trying to understand, the connection to function, and then the connection to healthcare. Not just developing the drugs, but understanding which drug to deliver to what patient. So modern medicine really began right here in Greece with the Hippocratic Oath and this ability to now do no harm and try to understand patients and patterns, and then use these patterns of intervention to treat patients, even when we didn't really understand the mechanism underlying many of these diseases. This changed dramatically with new technologies. So the technology of the microscope allowed us to now look inside the body and understand the cellular basis of human disease. And as early as 1911, Alois Alzheimer diagnosed the first patient with Alzheimer's disease using microscopy by looking at 2 phenotypes in 4 cells in each particular case, and then using that to recognize neurofibrillary tangles and amyloid plaques that are, to this day, the way that we diagnose Alzheimer's patients. So really not much has changed on that front in the last 115 years. But what has changed is this extraordinary availability of data. On one hand, we have genetic information. Genetic information allows you to go from correlation to causation, to understand that there are hundreds of genetic loci across the genome that are contributing ever so slightly to Alzheimer's disease. The vast majority of genetic loci, because they're constrained by evolution, are only making very tiny changes to disease phenotype. If they made bigger changes, evolution would keep them at such a low frequency that we wouldn't see them in the population. So by definition, almost, common variants are a very weak effect. And that's where AI comes in. That's where the ability to understand disease from a systems perspective, from a global perspective, to build these virtual twins, these digital twins that we've heard about, this ability to now predict the impact of all 6 million common variants and your thousands of rare variants and how they're acting in your own genome. So that's what we're up against. On one hand, 3.2 billion letters of ACGT that we're trying to really comprehend. Number 2, all of the cell types, the thousands of cell types in the trillions of cells of the human body, and in particular for the human brain, which might be the most complex entity in the universe, sitting in between your 2 ears. So understanding how these millions of neurons are interacting in billions of synapses and interconnections between them, and then leading to human thought. Again, we marvel at what AI can accomplish. AI has made extraordinary advances, and we should celebrate that. But we should not forget that we are almost on par with AI. I'm not going to say who's slightly ahead, but we're very close to each other right now. But we run on 3 watts of power. To build a human brain, all it takes is sex. And then 9 months of gestation, and of course another 18 years of growth and development. So this ability to now start from a single cell, a single zygote, that grabs half the DNA from mom and half the DNA from dad, and then replicates itself to autonomously not only create a body plan, but rewire the entire brain, number 1, and number 2, retrain the entire brain at every generation with a tiny snippet of what ChatGPT required for its own training. So every one of us is faced with a tiny subset of human civilization and the knowledge that we have. So it is quite remarkable to sort of celebrate this. And in fact, here I'm showing you 2.4 million human brain cells at single cell resolution. So notice the contrast. At the bottom left, I'm showing you 4 cells and 2 phenotypes. At the bottom right, I'm showing you 2.4 million cells across 400 individuals. And every dot is a cell, and it represents a vector of 20,000 variables, the expression of every one of all of the genes in the human body. So for those of you who love complexity, there's nothing more complex in the universe than the human brain. And that's what we are currently studying, to understand how these genetic differences and how these gene expression differences and how these gene regulatory differences lead to the extraordinary diversity of human personality, of human thought, of psychiatric disorders, of neurodevelopmental disorders, of neurodegeneration, and so on and so forth. So for those of you excited about the human brain and using the machine brain to understand the human brain, this is one of the greatest challenges of our generation. Now let me tell you what we're actually looking at here. So again, I mentioned that every dot is a vector of 20,000 genes. Of course, the 20,000 dimensional vector, we can see 2, maybe 3, maybe 4 with color, maybe 5 with shape, but after 6 dimensions, we're kind of lost as humans. So what we need is ways to reduce the dimensionality into more meaningful axes of variation. Because maybe there's 20 genes that are all involved in the peroxisome, and another 20 genes that are all involved in mitochondrial, energetics, and another 20 genes that are involved in neuronal wiring, and so on and so forth. So every single one of these genes is acting in combination, and therefore you can't think of the 20,000 dimensional vector as independent variables. There's a much lower dimensional manifold where it lives. Everybody with me here? So these are the true axes of variation. These are the meaningful axes of variation. And this is what embeddings allow you to do. So instead of looking at every measurable outcome, we can instead look at the meaningful biological variables underlying this. And this does not only apply to biology and genomes, this also applies to human thought. We are now at an age where not only the Greeks, but the geeks are thriving, where we can now finally tell all our friends that we're basically studying philosophy and literature and history that we can do that too. All we need to do for the geeks is to take those documents and transform them to vectors. And then thanks to compositionality, we can add, subtract, divide, multiply, average, and we can write all of the summaries that you would like, just that our computer will generate them for you. So it is quite remarkable that by living in this embedding space, we can now understand human ideas and how they're moving in the context of optimism versus futurism versus war versus public good or military, and so on and so forth. So we can now start understanding the dimensions of variation of not only genes and proteins, but also documents and human thought. So that's what embeddings are all about. So this is the transformation that we've seen in the space of medicine, and the paradigms that have shifted that allow us to understand it are, number one, moving from hypothesis-driven research to data-driven research. Number two, moving from correlation to causation, thanks to genetics. And number three, and most importantly, I would argue, moving from classical data analysis to generative AI and deep learning. So instead of building a new methodology for every problem, where the human scientist does all of the thinking, we can now build foundation models, which are general purpose. They're trained using pretext tasks with self-supervision. For example, ChatGPT, you hide a few words and you say, predict the words that are missing. And the only way that it can get good at that is to truly understand human thought, to understand the human psyche, our hopes and aspirations and fears. And it has done a tremendous job at that. And the secret to that is these embedding spaces that I talked to you about. It is this representation learning. The fact that instead of computing on individual words or characters or phonemes, it is computing at a higher level of abstraction, at a level of meaning, where we can basically learn these hierarchical concepts that are then combined together, very often in multimodal ways between different types of modalities that I'm going to talk about in the next slide. And the AI can truly be our partner in understanding all that. So this is this latent space, this embedding space, this conceptual space that I keep talking about. So machine learning has traditionally been about X to Y predictions. We have a bunch of pixels, and we'd like to know if there's a boat, a car, a dog, or a person there. But those pixels themselves are kind of meaningless. And making this computation all at once has proved to be simply impossible. And machines have had a terrible time understanding pictures or understanding anything else for many, many decades. The key advance of deep learning and generative AI is representation learning. It is the fact that I can combine the pixels that are nearby together to form lines. And I can combine these lines to form shapes. And I can combine these shapes to form objects. And I can combine these objects to now recognize a car, a person, a boat, and so on and so forth. And the most exciting advance is that this is learned entirely de novo using self-supervision. And that lives in this representation space. So images is how the whole field got started. But of course, we have since moved to any type of modality. In the realm of DNA that I was telling you about earlier today, we can now understand the impact of individual mutations by studying the words, the meaning of DNA, the words that are recognized by proteins to bind there, and then the grammars that are built on top of these words. In the space of chemistry, we can now understand individual atoms and how they're combined, just like pixels, to form shapes. How they're combined to form much more meaningful properties. A single atom alone is not very rich in information, but when you put multiple of them together, then you get these rings and you get these polar properties and charge and hydrophobicity and so on and so forth. And all of these are emerging properties. They're at higher levels of abstraction. Is everybody with me here? Can I get a show of hands? Awesome, fantastic. Good. So we can do that at the level of individual atoms for chemistry and at the level of individual amino acids for proteins, where we can now combine multiple amino acids together. And we can do the same thing with patient records. We can now start understanding how individual patients relate to each other by building up information from their parts, understanding this reusability of components. And we can do, of course, the same thing with text. So you can understand individual words and map words using word2vec, map them into the embedding space. But you can also, thanks to compositionality, map entire documents. And then being able to do computation on those documents and summarization. And in the same way that you can understand chemistry using the context of one atom and the other atoms, you can understand knowledge graphs by letting every one of these nodes influence each other. And of course, even with raw numbers, you can still infer meaning from these numbers by understanding their covariation, which is what I'm showing you in this picture. And perhaps the most exciting part is, of course, the multimodality, the fact that you can combine all of this information together. So using this information now, we can start making new advances. For example, I mentioned to you earlier the structure to function problem. Well, my student Emma, who won a master's thesis award at MIT for her thesis, Emma Tiesinger, basically built this latent representation of every single protein in the human genome. And you can now recognize the functions associated with those proteins by understanding not only the structure, but also the function of those proteins, using these knowledge graphs and seeing how proteins are connected to phenotypes, how they're connected to diseases, how they're connected to drugs. You have genes here in the middle, and all of these connectivities that are reshaping the latent embedding representation. But you can now learn a joint representation between the protein structure and the protein function. So you can use that to now bring a much more meaningful representation to proteins, and you can combine that with chemical representations. So you can take every single chemical, every single drug. There's 11,000 drugs that have patents associated with them, that have FDA approval. And there's about 2 million drugs that have patents associated with them. And there's 121 million drugs total. So we have basically taken the landscape of chemistry and mapped it through these techniques to understand how drugs relate with corresponding proteins and how they're influencing each other. So we can also use that to understand, of course, patents and clinical subjects, and now relate the drugs on one floor to the proteins on another floor. And we can co-embed them based on the proteins and drugs that are bound to each other. So using these Morgan fingerprints for the drugs, using T5 for protein representations, and using domain representations as well as patents and their co-embeddings, we can now build these joint representation of drugs and proteins. And we can use that to, of course, study individual drugs, but we can also study here the 121 million chemicals that are in PubMed. So that allows us to now start not only embedding these chemicals, but also we can start outbedding. We can start generating molecules from the latent space by understanding the regions where specific diseases are richer. So here's 3 drugs, for example, and then here's 3 target proteins, and then the drugs associated with them. And here's the co-embedding of these drugs and proteins together with experimental evidence that you can see matches the prediction that we have from our model. And here's the ability to actually generate new drugs by sampling that latent space. And you see that the space is actually smooth, that you can actually cluster proteins and chemicals in a meaningful way near each other. So that allows us to now start taking on drug discovery using these latent representations. We can now start understanding how to use all of this information across all of these different modalities and building agents that are playing the role of different employees or different leaders inside your own company. I can now understand gene expression patterns using specific tools and understand how to validate specific targets and understanding how to use the modules, how to obtain tissue expression, how to then read the literature across the entire body of literature that humans have ever written to understand the function of those target genes and how to select drugs using this approach. So you can now ask it, hey, find a combination of approved post-phase 1 clinical trial drugs that can treat Alzheimer's. And it goes through all of these capabilities and then it predicts drugs. And it's able to now carry out, of course, the generation of the molecules using the latent embeddings that I showed you. But it's also able to use biophysical docking to now infer the binding affinity of these molecules. And my entire team was blown away by the affinity values that we're finding that are, the dream of pharmaceutical companies to obtain such compounds at such a large scale. So you can see here the different agents at work. You can basically see the reasoning of how these agents are thinking, how they're working, how they're loading up tools, and how they're basically being used to select targets, validate the targets, select drugs, and then ultimately generate a report. And we're now scaling this up to 121 million chemicals, as I mentioned. So we can use this along with geometric modeling, using geometric deep learning, to now start predicting new molecules for cardiovascular disease, for melanoma, for Alzheimer's, and using this to now start guiding these reasoning, interactive, user-guided, and AI-powered drug design platforms. And in the space of cells, instead of thinking of every cell as having a binary phenotype, where every cell from an Alzheimer's patient is 100% Alzheimer's, and every cell from a control patient is 100% control, we can now use this to map much more refined phenotypes within the embedding landscape of the expression of every single one of those cells. So using transcriptional similarity in this embedding landscape, we can now position every cell. And using the phenotype of the donors, we can now color every cell. And we can then find neighborhoods that are very Alzheimer's and neighborhoods that are very controlled. And we can then go and recolor the cells to basically say that even though this patient is diagnosed with Alzheimer's, all of their microglial cells are sitting in healthy neighborhoods, but their oligodendrocytes are sitting in sicker neighborhoods. So we can use that information to now start revisiting the phenotype of every cell, and then of course the phenotype of every patient by sort of going through and for every cell type, looking at whether this patient has healthier glial cells but sicker neurons. And not just for Alzheimer's, but also for tangles, cognitive impairment, amyloid beta, neuroinflammation. For every phenotype, we can now project it at the cellular level. And that gives us extraordinary advances in being able to detect differentially expressed genes and being able to detect subtypes of patients. And that allows us to now start mapping within these latent landscapes the patient trajectories directly. So that allows us to now start rewiring the circuitry of human cells. In this particular case, we basically showed that in the case of obesity, we're able to uncover the mechanism leading to the storage of fat in our cells. So 42% of European chromosomes carry a risk allele for this particular variant that we showed is in fact leading to storage of calories instead of burning of calories. But by flipping a single letter out of 3.2 billion letters, we're able to restore thermogenesis. And by changing the expression of a single gene, we're able to now restore a healthy body weight in mice that are simply unable to gain weight in a high-fat diet. So that's what we want to do systematically. And that's where this representation learning comes in. So in closing, what I want to leave you with is the idea that we've made extraordinary advances in AI with deep representation learning, multimodal understanding, and intuitive reasoning. But by contrast, where I think the field took a wrong turn is that our interactions with AI are now only based on language. And we're treating AI as a black box with no peeking inside the box. And the humans are left outside the loop. And that's why we developed Mantis, this AI-powered, human-driven, interactive, visual data science workbench that allows us to now take these 20,000 human proteins and map them into their latent space and start interacting with them using the lasso tool or using search or by studying different neighborhoods, understanding the ontologies, being able to plot properties, being able to ask the agent and the orchestrator questions about any part of the space, and being able to see the thoughts of those agents mapped back into the latent landscape. And of course, being able to collaborate with multiple folks sharing their geolocation in this cognitive space. And we have developed a panoply of tools that you're able to use to understand, to extract knowledge graphs, to build agents, to be able to see these agents working directly in this landscape. So here you can see 3 million articles from the New York Times mapped onto the Mantis cartography. I would love to take every Greek law, every politician, every company, every person, every scientific paper, and map them into this cartography. And I want each of you to think, hey, what would I map if I had access to such a tool, which by the way you do. And I'll be sending everybody a link to this. But we've used this to map every scientific paper written by MIT. So 60,000 papers here, every grant, and the 2 floors connecting the scientific papers and the grants together, but also the physical place at MIT where their offices sit to identify new opportunities for collaboration. Here we can see the links between every course that has ever been taught in MIT or edX and the prerequisite relationships between them, allowing us to say, well, maybe, yeah, you can substitute this for that because, the links are still working. Here's every startup in PitchBook where you can basically see now combinations of properties of their founders and their performance to start building predictive models in every region of the startup landscape. Here you can see 100,000 loans and quantitative information about the FICO score for how they're working. So, and of course, this is multimodal. This doesn't need to be just about text. As I showed you earlier, we can now study protein structure. We can now study images. You can take pixels and map them into the embedding landscape. You can take chemicals and see how they're related to each other. And all of these are natively inside there. So that's the power of maps. Instead of just receiving a text message every time you need to turn on a ski lift, you can see the entire landscape. That's the power. And this is the architecture of Mantis. These are the 4 pillars on number 1, embeddings, number 2, knowledge graphs, number 3, agents, and number 4, of course, the tools that those agents are able to use. So you can basically, and by the way, the team that works on Mantis includes several young Greek students, and several of them are here, and we are still recruiting. The youngest member of our team is actually 13 years old. He just turned 14 yesterday, so we're looking for a new 13-year-old wizard. But all I'm trying to say is that there's no limits to what students can accomplish nowadays. And I want every young person in this room to not only say, yeah, great, I want to use Mantis, but to look at this map and say, oh, I want to study embeddings, I want to study knowledge graphs, I want to build agents, I want to sort of agentify every tool inside Mantis, inside this MCP framework. And I really invite every single one of you to sort of find the best hackers you know and ask them to join this team. Or if you are the best hacker you know, you should join that team. So we have an extraordinary set of capabilities. I'm not going to go through them, but we have all of these data science primitives, we have all of these quantitative data analysis, all of this in-place visualization, we have this collaboration where humans can co-browse with each other, where they can leave geotag notes. We have all of these agents that are able to reason based on what you're asking them and write code and execute the code. We have a notebook that actually is able to, you ask it, hey, I'm wondering, how do the number of likes correlate with the number of, words or the specific words inside every Lex Friedman podcast? And it analyzes these millions of points in seconds and reveals insights, and is able to now code both front-end and back-end interfaces, automatically within this tool. And we're also able to embed Mantis inside your browser. So we have a Chrome extension, so that next time you browse Wikipedia, instead of browsing linearly, you can basically now take multiple articles. Here's actually the Wikipedia article for artificial intelligence, but you can find every concept in that article semantically, and then click on them and then see the corresponding paragraphs light up based on their actual content. And here's one example of our agentic workflows and how you're putting them together. This is actually from the hack that we carried out last weekend. And Nicolas Brigadas, who's actually filming right here, led this hack with the healthcare team, where you're able to now identify biomarkers and figure out which ones of them are causal and which ones of them are actually good targets, using thousands of papers from the literature and then this agora of collaborating agents. And you're able to now install Mantis in your own computer, your own backend. Here's the end user. You can use our front end, and you can use our own default front end, or you can use your own corporate backend. We have an NCP server and all of these agentic actions, and you can even register if you have a supercomputer that you have access to. You can register so that the notebooks are using that. So you can basically achieve versatility, because you can plug in any embedding model from ChatGPT, Claude, Gemini, and we have also our own LLMs. You can also achieve privacy by loading up your own local implementation of DeepSeek or Kimi K2 that just appeared yesterday. And you can also scale to billions of points using this progressive loading of points. And the other fun thing about Mantis is that we have used Mantis for Mantis. In other words, we have embedded every single conversation that our team has ever had. And we can now ask questions about, hey, where's Pascal working on? And what about Legion? And what about, team, D for embeddings or A for agents and so on and so forth. And you can now interact with this landscape. So that's where I'll close to basically tell you that the road ahead is one where we collaborate. We don't just leave humans inside the box. but we lift them up. The analogy that I like to give is the human being Aladdin falling in the cave of wonders, but locked inside a box. And the AI being the genie, able to fly anywhere and see the treasures. But Aladdin can only ask their questions in a little piece of paper. When the genie sees it, it sees all the treasures, but the answer is just written in a piece of paper. So we've built a magic carpet so that Aladdin can now go and fly and see the data directly. And that's where I think the future lies, where the AI is empowering this extraordinary visual acuity that humans have. Not just one answer at a time, but thousands or millions of documents at a time. And I don't want to just build a magic carpet, I want to build a F-18 control panel. So that's the two teams that are working on this. On one hand, we have the computational biology team, but on the other hand, we have this Mantis team of youngsters, and you can see the median age here is 17, and that's true across our entire team. That's where I'll stop, and I'll be delighted to take any questions.
So let me announce that we are looking for volunteers for Mantis, to use Mantis for the Greeks in AI, because actually what we are trying to do is a cartography of our great community, the very vibrant community that it is formed. So what you presented today is mind-blowing, it's amazing. Thank you very much for this so interesting talk. And let's start this QA session, some questions. Yes, yours.
Great talk, Manolis. Your data have a strong spatial structure and a neighborhood structure, and I noticed you use graph neural networks. So I wonder if you could elaborate on how important the structure is in getting the embeddings and all this.
Yeah. So a fantastic question. So the question is, how do you encode structure in a chemical? So basically what we do is that we have first a graph neural network that is able to bring information across all of these atoms together, and then through the message passing is able to then refine at every layer. In the first layer, you only have information from your immediate neighbors. The second one, you have the neighbors of your neighbors, etc. And eventually we use a variational autoencoder that is able to then combine all that together. Now, you're asking a fantastic question, which is, how much is the structure mattering? And in this particular case, one of the requirements that we had in building this affinity net, which basically recognizes chemical affinity, is that the reconstruction accuracy was actually quite high. So basically, this is what we call outbeddings. So embeddings go into the bed, outbeddings go out. So the outbeddings needed to actually match the molecule, and not just some vague approximation. But so that forced us to now have these very geometrically driven representations, and that led to extraordinary advances. So we are now the top performing team across the Therapeutic Data Commons benchmarks with version, a very early version of our model, and we're only continuing to increase. So yes, geometry really matters, and you can encode it using these geometrically constrained graphs, where the angles are actually mattering.
Thank
you. Congratulations from me as well, Professor Kellis. I would like to make a question about the basic hypothesis about the meaning being embeddings. Usually in much simpler situations, for example, when you're compressing information, the final result is too tight and is difficult to derive meaning that is humanly understandable. And sometimes human meaning itself carries a lot of redundancy. So I would like to ask, to what extent meaning is essentially something that we cannot see in the process rather than the embedding. For example, in variation autoencoders, we have the system, the decoder, that takes something that is, in a sense, meaningless for us, and it attributes it meaning through the decoding process, and we get a meaningful result at the end. So, can we enhance this understanding that we get from the embeddings by taking into account the meaning that comes from the process itself, from the weights, essentially, of the system that does the decoding?
This is a fantastic question. So basically, I want to start with an example. So this is every conversation that Lex Friedman has ever had. And at the center of this all lies, pertaining to your question, the meaning of life, interestingly. so next to the meaning of life lies death and meaning perspectives, existential thought, reflections and insights, philosophical discussions on life, advice to young people, and so on and so forth. So I have spoken to many people about their own data about this. PBS Nova had an episode on AI, and I was one of the participants. So they came to my office, and as they were taking the elevator up, I told my students, hey, why don't you build a space for every Nova episode? So they went, hacked it, downloaded it all, and created a space within a few seconds. And then I showed it to them, and they were spooked. They basically said, I feel seen, because the way I think about these episodes is the way the AI thinks about these episodes. And I think this is very profound, exciting, and perhaps troubling. But it is perhaps no surprise that all of us understand each other, even though we have only read a tiny subset of the world literature. It's because we learn through association, and ChatGPT also learns through association. So the embeddings that it creates are not some random embeddings. They are the embeddings that you get from our own corpus of data. And it turns out that you need a tiny subset of that for humans to be able to understand each other. If I tell you to use the words liberty or freedom in a sentence, you might think of them as synonyms, but you use one or the other because of the context. So the other experiment that I want to share with you is that when we ask the agents questions, if we only give them the original text, they answer one way, but if we give them the original text and the hierarchy of clusters that leads to that document inside the corpus that we're looking at, they bring such a more meaningful interpretation. And the last thing I want to say is that there are axes of variation in that latent embedding space. That there's an axis for futurism, there's an axis for humanity, and there's an axis for historical values, there's an axis for war inside the Lex space. When we studied the dimensions of variation, not in the 1,500 dimensional space of the embeddings, but in the 20 dimensional space of the compression, those were so meaningful. And we can now start asking, okay, if I tune this, what are the outbeddings that I get? We're able to generate new documents by pushing out different functions. So I just want to say that the meaning that emerges has blown us away time and time and time again. And like all of these experiments are telling us that we're onto something very special here. It's not just a fun visualization. We're truly getting at the thought inside the brain of the machine, which might not be that different from the thought inside our own brain. And there are experiments, there's new scientific papers where scientists have studied the firing patterns of the neurons of humans looking at different concepts and the embeddings of LLMs looking at the same concepts, and they find an isomorphism between the two. And there's more and more research that shows that the embeddings that ChatGPT has and Claude has and DeepSeek has are in fact making a similar map of the space. So I'm kind of optimistic that there might be a truth out there, there might be a meaning, there might be what our ancestors like Plato and Socrates were looking for, and we now finally have maps for them.
Hello, hi. Yes, my question is quite similar to our fellows. Sometimes I have wondered if we sometimes over-rely on the existence of latent variables, and apart from the practical implications that we see, how effective they are, my question is how do you think about the ontological status of these latent variables?
So I hate ontologies. Ontologies were built by a bunch of old men with beards in the 80s. And somehow we still think that they're relevant today. So when we're thinking about psychiatric health for 16-year-olds in the age of AI and TikTok, what do they know? So what we have done is, mental health. So what we have done is that we ask young people, how do you feel? And then they answer just in a paragraph. We don't need ontology for that. And from that paragraph, we created a Mantis landscape that basically realize that there's work-related anxiety. There's parents getting older related anxiety that were not in the DMG codes that those scientists made back in the 80s. So I think that ontologies are obsolete as soon as the old men leave the room. But what we have here is the ability to create these ontologies completely from the data directly. And you can of course browse these. You can also look at let's see, View Explorer. So this is the entire ontology of the Lex Clips space, for example, at multiple layers of resolution, and you can kind of now see how they're capturing different parts of the landscape. So I think dynamic ontologies is the only way to go. I think all of these people trying to build an ontology that will surpass all ontologies. I don't know if you saw this XKCD cartoon, how standards are created. There are 14 standards for this. We need to create a standard that unifies them all. Scientists work. The last panel, there are now 15 standards. so yes, I think that ontology should be built dynamically and never by humans. They should just be built on the fly. And we also have generated tools that if you have a corpus from the New York Times and a corpus from, I don't know, National Review, you can kind of match the ontologies to each other. Because of the vector computation that we can do in these latent spaces, you can actually unify the ontologies. So I think that they should be just built on the fly, thrown away on the fly. Yeah, great.
Hello, I apologize, my question is going to be a bit less philosophical than the previous ones. So I want to go back to the structure to function prediction.
Yes.
And I'd like you to clarify a bit what's happening. So if I understood correctly, there's a subset of the 20,000 human proteins of which we know the function. And then what you're doing is essentially estimating the function for the remaining ones, for which we're not sure about the function. And the question is there a way of actually evaluating these predictions, especially considering how complex these gene interactions really are?
Yeah, so it's a fantastic question, and I have to admit that was not our goal. Our goal was not protein function prediction. Our goal is to now go back to understand all of human biology. When I have the expression vector of those 20,000 genes, as I showed you in my first slide, those 20,000 genes are meaningless. They're just a bunch of numbers. The IDs of these genes are meaningless. They're just a bunch of numbers. So what I want to do is bring meaning to them all. And the reason why I'm worrying about this is not that, there's a few genes that we don't know the function of. What I want to know is, what are the amino acids responsible for the function? So when I say structure, I don't mean, we have the whole structure of the protein. I want to know that this gene has 17 functions, and this one requires those 4 domains, and that one requires those other domains, etc. So basically, again, this is like Emma's thesis directly, which is now trying to do this directly at the domain level. So we're able to now take, domain representations and infer meaning for them. So the evaluation is not, is this protein a transporter? I'm pretty sure it's a transporter. In other words, I think those benchmarks are very archaic in my view. I think that we're well beyond that. What I care about now is, what are the domains that are responsible? Because every protein is so pleiotropic. We found expression patterns of immune genes inside neurons. And these are clearly annotated as immune genes, and they're known as immune functions. So I think that the annotations that we even have for these genes are very archaic, and therefore any kind of benchmark is going to be flawed just because of the very little information that we had when we first annotated those. But I think that the ultimate test for me and for my group is targeting these domains pharmaceutically and then showing that I can manipulate that function. And our current results have blown us away.
So thank you very much.
You sure you wouldn't want to stay another few hours?
Actually, we could, I think. But now we'll have a break, coffee break. I think that Manolis is going to stay with us for a little bit more. So let's applaud Manolis for this very inspiring.
make two very quick introductions. One of them is members of the Mantis team that are here. So we have Panagiotis Vlasis, we have Nicolas Pigadas, and we have Christos Vlasis are here. But we also have a very special person, which is my mom, who's here with us today. So